Hello, good afternoon. Today we're gonna basically wrap up lecture 7. And then I think that will be it. I don't think we need to start on number 8. Just yet. Maybe I don't know. I'll see how long it takes to finish 7. Okay, if we have some time left, maybe we'll start on 8. But last time we were talking about the process of transcription and then translation, and we just want to take a closer look at what's happening with the translation today. And we're also going to learn about mutation as well. So hold on, let's see where we are. Okay. Here are some questions. This is where we left off last time, I believe. And here is poll question. If a codon contains 4 nucleotides instead of 3, what is the maximum number of amino acid such codon combinations can code for? So the way to do this is basically, think of 4 blanks instead of 3, and we would have 4 choices for the first blank: A, C, T, G. Same thing for second, third, and fourth, and you multiply all of them together. Okay, so that would be 16 times 4, which is 64. 64 times 6 is going to be 256. Okay. So when it comes to translation, there are different types of RNA that are involved in the process. We already know about mRNA, for example. That is the messenger RNA, which is a copy of the gene that we're trying to make the protein from. And it's a copy of the DNA, right? So we did the transcription to create the messenger RNA, and we did some RNA processing, right, to make it ready. So we took away the non-coding introns. We glue together the exons. We also stabilize the mRNA by adding a 5' guanine cap, as well as a poly A tail, a bunch of adenines on the 3' end. And all these features help stabilize the mRNA in the cytoplasm so that it's not going to break down as easily. Right? So you can think of the mRNA as the recipe, as the recipe to make the protein. Right? And then we have the rRNA, the ribosomal RNA, which is the type of RNA that help mix up the ribosomes. You might remember ribosomes are the organelles which are responsible for protein synthesis, but the ribosome themselves is made up of a large and a small subunit that consists of proteins as well as rRNA. So the rRNA is the one, the ribosome. It's the one that is going to be decoding the message on the mRNA and then physically connecting the amino acids together. When you connect the amino acids together, you're forming a peptide bond, right? So the mRNA is like the recipe. The rRNA is like, the chef that's cooking the recipe. And then we need something to bring the ingredients along, right? To bring the appropriate ingredients to the ribosome. And that will be the job of the transfer RNA, tRNA. Right? It's going to be bringing over the amino acid that corresponds to the codon. Right? If you see AUG on the messenger RNA, that would be the start codon. Then the tRNA is going to bring over the methionine, right? The MET amino acid. So these 3 things work together to make it possible for protein synthesis. Now, let me just give you a brief description of what these things are kind of look like, and and hopefully it will make sense to you. Okay, so let's just take out a piece of a paper, and then we're gonna quickly draw some sketch here. So let's start with the let's say we have the transfer RNA. Let's understand what the transfer RNA is. Okay. So the transfer RNA looks something like this. Okay. It's going to be like a clover leaf structure. First of all, it's RNA, which means it's single-stranded. But this single-strand RNA, it's able to kind of loop back on itself. Yeah, it's just folding back onto itself and form this clover leaf structure. Okay, so it's 5' and 3' here, and sorry, maybe let me draw this a little bit better. I mean, you don't really have to erase it. I just need more space is all. So it's more like this. And more like a clover leaf structure like that, okay? Now, I'm totally making this up. Let's say this is ACGU, and then on the other side, it would be UGCA. So it's able to kind of stabilize its structure by forming these internal complementary base pairing. Okay. So complementary. Yeah, go ahead, Nika. Am I pronouncing it right?
Excuse me, Professor,
Yeah.
why the 5' is short and 3'? It should be like this or no? Just
Yes, it should be like that. And I'll tell you why in a second. Okay,
okay,
very good
okay.
observation. Yeah, she's asking why this is like it's kind of down here and not all the way up, right? Okay, very good. Okay, and same thing here. You're gonna have more like complementary base pair within the same strand. You understand that this is where's my oh, I lost my pipe cleaner. But it's just single strand, okay? But then it's able to like loop back on itself, okay, and form a loop structure, and then the base pair here happens to be complementary, and it stabilizes it, okay? So there we go. And over here at the 3' end, the reason why it is like a little bit longer is because this is where it will be attached to an amino acid. Okay, so this is amino acid attachment site. Okay, amino acid attachment site. Now, at the bottom, remember this whole black line here. It's the whole black line is a RNA, right? So it's the whole thing is made up of A, C, G, and U. I just didn't draw all of it, but like I could, right? I could put like A, C, C, U, U, so on and so forth, right? It's gonna go all the way around. Now at the bottom of the loop, there is gonna be a triplet that is particularly important, right? And I'm just gonna put UAC like that, UAC. So again, the whole thing is just going to be, random stuff. You don't have to write it if you don't want to. Okay. I'm totally just making these things up. It doesn't even, have any meaning to it. Okay. But what does matter is going to be this triplet down at the bottom of the tRNA. This is called the anticodon. Okay, anticodon. The anticodon is going to be complementary to the codon found on mRNA. Just copy that down first, and I will explain to you what that means. Right? Just copy that down first. And I'll explain. Okay, so here is the messenger RNA. Messenger RNA. We're gonna have the 5' and the 3' end. Okay. Let's just say at the beginning, you know what? If let's make it a little bit shorter. People are gonna get maybe confused if it's not at the beginning. So the start codon, as you know, is AUG, right? So I'm gonna put a start codon right here, AUG. And AUG, if you look it up in the codon chart, please look it up if you have the codon chart. What does AUG code for? Can anybody tell me what does AUG code for? You know, this
MET.
what is that? I'm sorry.
MET.
MET, that's correct, right? So AUG codes for MET. So this amino acid has to be MET. You see that? The tRNA, the anticodon has to match the codon for it to come over. Now, I know this is a little bit confusing at the moment, but stay with me, please. Let's say I have another one here. Okay, let's say this one has the UUU on it. Okay. If it's UUU, can you tell me what this is? What would the amino acid be if it's
PHE.
UU? Okay. That is a good try. Okay. Anyone else? Anyone else?
PHE.
I'm sorry, what's that?
PHE.
PHE. Okay, so I hear people say PHE. Anyone else? Like if the anticodon is UUU, what amino acid is it going to be carrying?
MET.
Okay, I have PHE, I have MET. Anyone else? I'm talking about this one right here. If the anticodon is UUU, what is it going to be carrying? Now, I know many of you looked up UUU, right? And you say PHE and you don't understand like why is it not right. But remember, this is the anticodon. If it's the anticodon, what is the codon on the messenger RNA? What would the corresponding letters be? It'll be AAA, right?
AAA, right.
AAA, that's right. So we actually look up AAA on the table. And AAA codes for LYS. Okay, so that's why this is this. All right, now stay with me, listen to me, and see if this makes sense. So I'm gonna go back here for a second, okay? I'm gonna make this up. You don't have to copy. You can just watch and try to understand, and then you can watch the recording later to copy it, okay? So let's say we have AUG. I'm making this up. Okay, CCA. GCG, and then UUC, and then just one more, UGA. Okay, so I'm gonna have a bunch of tRNA here. Okay. Maybe not a bunch of them. Let's just do, 3 of them for now. Okay, so they all have their anticodon. Okay, so just pay attention to here and see. Now I have 3 tRNA here, and each of them is going to be carrying an amino acid. Okay. Now we'll talk about what those amino acids are later on. So let's say we follow this one. Okay, this one has an anticodon of GGU. Right? So it's going to be floating around, it's going to try to come to the mRNA and it's going to try to fit here. Okay? Is that going to fit? Yes or no?
No.
No. Why not?
Because G is not complementary to A.
Exactly, right? So this is not going to fit. So the amino acid that it carries is not gonna come to the messenger RNA. It's just gonna bounce off. Okay, and then the next one you're gonna say, okay, does it fit? Well, again, I hope you can tell me that it is not gonna fit, right? Because then the red letters are not complementary to the black letters. The anticodons are not complementary to the codon. So this one is also gonna float away. And then this one, is that gonna fit?
Yes.
That one fits, right? Okay, so it's gonna stay there and it's gonna leave the amino acid. So AUG, we look up the codon. We never look up the anticodon. So AUG, we know it's MET. Okay? Now can
Sense.
sure.
So where does this anticodon come from?
Okay, so the whole tRNA is AUCG, yes?
Yeah.
At the bottom of it, like when it folds back on itself to form this clover leaf, that's what it's called, right? Clover leaf.
Okay.
At the bottom of it there, it's a triplet. That's particularly important. And that's called an anticodon. It's part of the tRNA.
Oh, it's always part of it.
It's always part of it. It's always at the bottom.
Oh, okay, thank you.
Yeah. So the anticodon,
Sorry, Vincent, sorry, I have a question. So whatever you put in the under the tRNA is part of that one, right? Part of tRNA.
I'm sorry, one more time. I didn't quite catch the whole question.
This RNA that you put under the shape of the tRNA is part of the bottom part, right?
Yeah, so this is the bottom part of the whole tRNA.
And the underneath is that part, right?
Underneath where? Sorry.
This you made it bigger, right?
Yes, I just made it bigger. That's right.
Okay.
So like like this here is just that part, right? Does it make sense? Is that what you're asking me?
Yes.
Okay.
Yes.
All right. So, can you please tell me what this one is carrying? Like what's the amino acid for this one? If the anticodon is CGC, what amino acid is it going to be carrying? Again, you need your table. All right. And we are not looking up CGC, right? We never look up the anticodon. You have to change it back to codon to think about it, right?
VAL.
Is it though? Is it? Let me see. Which one did you look up? I'm just curious, if you don't mind.
I think it's GLY.
GLY. Okay. Let me just put the chart here. I think it's better if I put it on the screen. Okay. So this, I'm just gonna bring this guy over. So sorry, not enough space.
I have ALA.
ALA, yes. Okay, look, guys, this is anticodon. So we must figure out what the codon is, right? Like what is it gonna bind to on the messenger RNA? So what's complementary to CGC? GCG, right? So we look up GCG, which means this is going to be ALA. I know some of you are still confused, but can you ask me some question like, what are you confused about if you are confused? What are you confused about? And how can I make this better? What's the question?
Professor, excuse me, we cannot use the red one in the table, just we should
That's right.
find
The red one is the anticodon, right? This is not the code. This anticodon, the purpose of the anticodon is to allow the tRNA to see if it fits into the messenger RNA. Like, is it bringing the right amino acid? Okay, so that's why we should always look up for the complementary one. Okay, let's try one more. Okay, what is this? What is this? GGU. What does G?
I have a question, Professor.
Sure, please go ahead.
When you select for G, we can you for example, for C we have a G, we have a C and G, yes, for ALA,
For ALA, yeah.
yes, and then why we take this one GLY?
Why
Why
we
we-
take this one for what? I'm sorry.
For example, we took ALA, yes,
Yes.
and instead of ALA,
Uh-huh.
why we didn't take GLY?
Why we did take GLY? Okay.
Yes.
So this here, right? CGC is the anticodon.
Mhm.
So we look up GCG. Do you agree we look up GCG?
Yes. Yes.
Okay. So G, let me just take my highlighter out. G is right here.
Yes.
Okay. And then C is right here, right?
Yes.
So it has to be one of these ones, right? And
Okay. Yeah.
and then G. So like right there. Do you understand?
Yes.
Okay.
Thank you.
Yeah. So what about this? Okay, this is GGU. What's the amino acid that is going to be carrying?
PRO.
I'm sorry, what was that again? PRO, did you say?
Yes.
Yeah. Can someone confirm that? Or you have a different answer? What's going on? Let me know.
Yes, PRO.
Yeah, because we look up CCA, right? So CCA. So that's going to be PRO. Okay.
Vincent, these folded ones are tRNA, right?
That's right. The folded
So
thing is
which
TR.
one? The bottom one is mRNA, right?
This black one right here?
Yes.
Yes, yes.
I mean all the strand. Yes.
This one, right?
Yes.
Yeah,
Yes.
that's the mRNA. Yes.
Okay. Thank you.
Okay. Now, so listen, right? Now, we the tRNA, I should label it as well. These are tRNA. I know it's very hard for you to copy. Okay, this is not meant for you to copy. This is meant for you to kind of understand what's going on. Now, this one is gonna come, for example, it's gonna come here, and it doesn't match, right? It doesn't match because CGC is not complementary to CCA, right? So this is just gonna like go away, which is good because CCA is coding for PRO and not coding for ALA, right? So this one's gonna come buying here, and then the ribosome, which I haven't drawn yet, but there's gonna be the ribosome right here, the ribosome. The ribosome is gonna now connect the two together. It's gonna make a peptide bond. Okay, it's gonna make a peptide bond. And then the tRNA is gonna leave. It's gonna go back to the cytoplasm. Okay. So now the tRNA is empty. It doesn't have an amino acid anymore. But in the cytoplasm, there is like a bunch of amino acid, right? So the tRNA is gonna have to like reload. It's gonna have to pick up another amino acid, but it has to be the correct amino acid, right? So the anticodon is in a way do two things. Number one, it makes sure that the tRNA is gonna pick up the correct amino acid, and number two is gonna make sure that you deliver it to the right place. Okay, and then for the next part, right? GCG. You're gonna bring this one over, right? And then the ribosome will form another bond. And now this one is gonna leave. Okay, and then the whole ribosome shifts forward, and you keep on building and building. Okay? So I know that's really hard to visualize, but let me show you in the PowerPoint here.
So Vincent, one ribosome is enough for building up a protein molecule?
Yes, one ribosome molecule is enough to make a protein, but you have like a lot of these ribosomes all working together, right?
Yeah, thank you.
So here, number one, amino acid and tRNAs flow through.
Excuse me.
Yep,
Can I ask a question?
sure, of course, yeah.
So at the bottom of the tRNA is the template.
At the bottom of the tRNA is
Is
the
it the-
anti-codon. Yeah. Are you asking me if this is the template strand? Is that what you're asking?
Yeah.
Okay, so
Yeah.
Okay, I understand where you're coming from, but don't confuse yourself like that. The template strand and the leading strand is in the DNA, right? It's in the DNA. Now we're talking about the RNA. Okay, but you're right. The anticodon is the same as the template strand in
Yeah.
terms of like the letters, right? In terms of the nucleotides. Okay, that's very good. But the T becomes a U, right?
Yeah, I see it as a template, and then I need to change it to the eRNA. mRNA.
That's right.
mRNA. Okay, I got it. Thank you.
Okay. So back to here. Now we have amino acids floating around. We have tRNA floating around, and some of them are empty. And the reason they're empty is because they probably just deliver the amino acid to a ribosome, right? So there's going to be an enzyme that's going to help connect these things together. Okay. So tell me, this one has an anticodon of GAG. Which one is it going to pick up? Is it going to pick up the LEU or is it going to pick up the SER? LEU or SER? Look it up.
LEU.
It's going to pick up the LEU, right? Which letters did you look up?
CUC.
That's right. We look up CUC, and then we figure out it's LEU. So the LEU is gonna get loaded onto this one. Okay, same thing here. You're gonna load up the SER onto here, right? Are we all clear on this? The relationship between the anticodon and the amino acid that it carries? There's like an extra step that you have to consider, right? Okay. Then you can see here is the ribosome. The purple thing is the ribosome, right? The top part is the large subunit and the bottom part is the small subunit. And so the tRNA will dock with if the complementary RNA codon is present on the ribosome. Right? That means like, these things will come and try to fit in, but not everyone is going to fit, right? In other words, the anticodon must be complementary to the codon. The anticodon on the tRNA must be complementary to the codon on the mRNA. And then you will join them together. Right? This one, where is this one going? This one is gonna reload another amino acid. Okay. Excuse me. And then you have more things coming in. And then the ribosome just gonna keep on moving forward and forward. Okay. So the messenger RNA is really long. I don't know if you can see my wire here. Pretend this is the messenger RNA. Okay. And then the ribosome will kind of hop on like my mouse. And then it just like keep on moving along and read the entire ribosome like that. Read the entire messenger RNA. It just keeps on moving forward. When does it stop? It stops when we get to a stop codon. Okay. When the ribosome reaches the stop codon, there is no tRNA corresponding to the stop codon. Okay. The stop codon, as you know, is UGA, UAG, UAA, right? There is no anticodon matching those things. And then the ribosome cannot proceed. Nothing's coming. And that pause is a signal that tells the ribosome this is the end. It's done. Okay, so everything falls apart, and then you release the protein. Okay.
So, Vincent, this happens in the cytoplasm, right?
It can happen in the cytoplasm. Yeah, okay, yes. Yeah,
But
so
where tRNA come from?
it's
Is
already in the
nuclear-
cytoplasm. Yeah, it's already in cytoplasm. Yeah, it is already floating around there. Yeah. If you want to know, like, are you asking me how do you make the new tRNA? Is that what you're asking? Or
Yes.
yeah, they are also made from the DNA. Yeah, but we didn't talk about it. I guess what I was trying to say though, like yes, it happens in the cytoplasm, but some happens like freely in the cytoplasm. Others happens in the rough ER as well, right? But yes, generally happens in the cytoplasm.
Thank you.
And then that's it. Okay, I'm not gonna ask you to describe this process, right? But you know, hopefully that makes sense. Okay, so here we go. The tRNA with anticodon CUU will be carrying. I hope you guys all know this by now. Okay, you just look up GAA. Right? Look up GAA, and what does GAA code for? GAA.
GLU.
Yeah, GLU. So that's it. That's the answer. Okay. All right. So I'm gonna go back to the study guide here for a second. And I'm gonna go to this table here. Okay. And I wanna do a few examples with you. I'm just gonna copy this over here, and I'm gonna bring my bring my thing down here. Okay. Now, hopefully you have your study guide with you. Otherwise, it's rather difficult for you to follow. So this is the coding strand. The coding strand is a strand of a DNA that contains the instruction to make the protein. Right? That's the what the coding strand is. So let's make the messenger RNA. Right? With the coding strand, how do we make the messenger RNA?
Identical.
Very good. Right? But the T becomes the U, right?
Yeah.
Yeah. So we're gonna go AUG, AUA, and so on and so forth. You basically just copy, but you switch the T to U, right? Switch the T to U. So that's the messenger RNA. And we're gonna process this thing, let it go into the cytoplasm, and then the tRNA is gonna try to bring the correct amino acid, right? So what would the anticodon be that would match this? What's the relationship between mRNA and anticodon? Is it identical? Is it complementary? What's the deal?
Complementary.
Yes, complementary. So we're gonna go like, you know, UAC, UAU, GCU. Okay, please check my work. I don't know if I'm doing it right. And then this one is like none, right? Remember, this is the stop codon, so there's like none. There's no anticodon for the stop codon. That's why it's gonna stop. Is this okay? Does anybody have any question about how I come up with the answer here? Okay, what about the amino acid? Do I look up the blue one or do I look up the red ones?
The blue one.
The blue one, the codon, right? Always look up the codon. Never look up the anticodon. So you just look it up. MET. All right, look up the AUA. AUA is ILE. CGA AAU. GAT. GAU rather. And then stop. That's it. Okay. Any questions at all?
Yes, Professor.
Yeah, go ahead, please.
The stop codon was only three, yes? UGA, UAG, and UAA.
That's right. So because it's
So, well,
UAA, we stop. Yeah,
oh yeah,
yeah, UAA, right? Mhm.
UAA, yes.
Okay, so I want you to try this next one, please. Okay, try this next one. And you can, when you have it, can you message me? Like you can private message me, right? If you don't want a class to see, or you can just let the class to see. It doesn't really matter. It's like your personal preference, okay? Try this out and tell me what you get. Okay. See if I can make this bigger. Okay, this is bigger now. Okay, remember this is template strand that I'm giving you right now. Template strand. Remember, you can send me your answers. I would be happy to look at it. Okay, if you're watching this on YouTube, you should be trying it out as well. Or you can skip forward and find the answer. Yeah, we gotta fill in the whole thing though, right? All the way to the amino acid. Send me your amino acid if you could. All right, I'm gonna write down the mRNA for you. Let me know if you need me to explain why it is the way it is. Now the anticodon is you can do it complementary again, right? But as Jing pointed out earlier, the anticodon is actually same as template, right? So that will save you some time. Okay. And we look up the amino acid. Does anybody have the amino acid sequence here? So I don't have to look them up. Anybody have the amino acid sequence to share?
SER.
The first one is SER.
Yes.
Remember that you have to look up the mRNA.
Okay.
Yeah,
Okay.
not the anticodon. Never look up the
MET.
MET. Yeah, and then the next one.
PRO.
Mm-hmm. Okay, thank you, Nazrin. Can I believe your answer? Or should I check it over? I think you're correct. Okay. I think you're correct. I just did a quick scan here and it looks correct. All right, that's it. Any questions before we move on to even harder stuff? That's how we make protein. That's happening like right now in your body. Okay. So, I don't know if you guys know this. I just realized not everybody is aware of this, but you guys 202, and when you go to the so there's the lecture archives class note and then the study guide answer. When you go to the study guide answer, that's the answer, and then actually there is the video as well. So the video is me taking up the study guide, right? So if you have trouble doing the study guide, as in like the questions we just did right now, you should watch that as well. It's gonna help you, understand more, hopefully. All right, that's it. We're gonna move on to the next part of the lecture, which is mutations. Right? Mutations. So mutations, we briefly talk about it when we discuss, the oncogene and then the tumor suppressor gene, right? These are permanent changes in the genetic sequence of an organism. Permanent changes in the DNA specifically. And changing these genetic sequence might result in changes of the amino acid sequence. Okay, the key word here is might. It's not 100% sure that you're gonna change it. Okay? As we'll see why. And you probably might have a good guess as to why that is the case. And we know the protein function is dependent on the amino acid sequence order, because the primary structure is going to determine how the protein folds up It's going to create its 3D shape. So if you change the sequence, you might end up changing its shape, and then you might end up causing the protein to function differently. Right? For example, we might cause a protein to be always turned on, like the green lights. And that's going to cause uncontrolled cell division, right? So changing the amino acid sequence is going to have a consequence on its shape, and that might ultimately result in a different function. Mutation can occur during any stage of the cell's life. It could happen in interphase, it could happen, even when the cell is not dividing, but it's particularly likely to happen when it's copying its DNA. Remember, copying DNA is a process that involves copying billions of base pairs, right? Even with the best spell checking and error checking mechanism, it's inevitable that if you do it enough times, mistakes are gonna happen. So I want to go through different types of mutations with you. Let's start with what we call base substitution. Okay? Base substitution is a type of point mutation. They are the same thing, okay, for our purpose. Point mutation is base substitution. And you can have three outcomes here. Right? It can be a neutral mutation. It can be missense or nonsense. Let's talk about neutral or silent mutation first. Okay. So I want to remind you that the mutation happens in the DNA. But then when you change the DNA, the messenger RNA is going to be different as well, right? And to make it easier for you to understand, I'm just going to start by explaining to you in RNA form. But keep in mind that the mutation happens in the DNA. Let's say we have UUU. This is the original codon. We call this the wild type. The wild type, the original. Okay. So then let's say it's mutated. Okay. This is called mutant. And it happens to become UUC. Okay, can you look them up, please?
UUC.
Yeah, what does you? Let's look up UUU first. What does UUU?
PHE.
PHE, right? Okay, good. This is PHE. What about UUC? All of you really need to do this to understand it. Okay, don't just take my word for it. Figure it out. What does UUC code for?
The
PHE.
same?
PHE, yeah. So look, if you have a mutation that causes the codon to go from UUU to UUC, that's a good thing, right? That's a mutation that's not going to do anything to you. Why? Because the amino acid did not change. It's neutral. It's silent. You don't even know you have this mutation. Okay? So harmless. Because it goes for the same thing, right? And that's why the redundancy is important. Okay? For example, like, there's a bunch of codon that codes for VAL. Right? So, the redundancy minimizes the effect of these mutations. Do we all understand why this is harmless and why this is neutral or silent? Okay, I have a question for you. I'd like to ask this question because most people have no clue. And then when they find out, they're like shocking. Okay. You and I, how much of our DNA do you think are the same? Give me a percentage. Okay. You and I probably are different ethnicity. Okay. Some of you might be same ethnicity as me, but for the most part, I think it's safe to assume majority of you are not in the same ethnicity as me. And we are not related. I don't think we're related at all. What's the genetic similarity between you and I then? Give me a number, a percentage. Type in the chat box. Yell it out.
I would say 0.
0, we're not, okay, we're totally different. No way we're. Love it. Okay, 0%. Hey, did you guys just look up the answer on the internet, huh? Some of you direct message me. Okay, yeah. Oh, with the right answer. Okay, some people say 90%. Okay, anyone else? Give me some more number. We have 0 and 90. Anyone else? Just maybe one more, two more. Okay, no more guesses. 50%. Okay, yeah. You know, it's shocking to many people, but between you and any other human beings on the planet, okay, not just between you and I, okay, between you and anyone else that you know, the DNA are 99.9% similar. Okay, 99.9% identical, I should say, not similar. 99.9%. Isn't that crazy? Right? Because we are both humans. Okay. So the difference, the point,
Vincent,
yeah.
About this, but is it not the chromosomes that makes the DNA? Yeah, and if people's, like chromosomes are from their parents, which are obviously like different people, like different continents, then like should it be this like the same?
Yeah, very good question. Okay, so the difference that distinguish one person from another is just that 0.1%. Because to build a human, you need the core material, core genetic material, and that makes up like 99.9%. Okay, meaning like we gotta have 2 eyes, we gotta have a nose, we gotta have 2 hands, 5 fingers, like all those core information, regardless of which continent you're from, what ethnicity you have. We all have the same stuff, right? So don't like you gotta have a heart, you gotta have 2 kidneys, so on and so forth. So all that stuff that makes a human is encoded within this 99.9%. That's why it's the same for everybody, right? And it's just the 0.1% that makes it different. I don't know if I'm answering your question. Am I? Or are you asking me something else?
I get you.
Right. So look, listen, right? So what makes you and I different is actually caused by a lot of these silent mutation. Okay, we actually call them single nucleotide. So just one nucleotide, single nucleotide polymorphism. A polymorphism is a detectable difference in the DNA between 2 people. So this is called SNP, SNPs. Okay, SNPs, polymorphisms. And this is important concept when we do biotechnology later. Okay, just one more question for fun. Yeah, sure, go ahead. Sorry.
So, for example, different person 99.9 DNA similarity, as you said. So what about sibling? Is that 100%?
Okay, so for that's also a good question, right? You've heard people say you are 50%, you share 50% of your genetic material with your sibling, right? Okay, that's probably where you heard it before. So we are talking about 50% of the 0.1%. So of the part that's different, the 0.1%, that's gonna be like really different between you and a random stranger. But for people who are related to you, then it could be 50%. But again, I'm talking about the 50% of the 0.1% difference, right? Okay. Does that make sense?
Yes.
Yeah. Okay, one last fun question. What about between human and a chimpanzee? Chimpanzee at the zoo. What percent are we the same? A percentage. If you look at the
50?
DNA. How many?
50.
50, yeah. Anyone else? Higher, lower?
98.
98, yeah. Okay. Now, you guys understand how the game works, right? Yeah, it's pretty high up, okay? For human and chimpanzees, 96%, okay? 96%. Remarkably similar, right? Again, it's like mind-blowing for a lot of people. Like, what? Like, we look so different, right? We're so much more intelligent than the chimps, right? And yet, genetically, we're only 4% difference. Can you imagine that? Okay. So anyway, moving on. Missense mutation. So let's say we have UUU. Okay. UUU. This is called wild type. Wild type. And then now we have the mutant. That's actually what they're called, okay? So UUU, let's say it mutates to UUA. UUA. Okay, so let's look up UUU again. We just did. Hopefully you remember it's PHE. And what about UUA, huh? What is UUA?
LEU.
Yeah, UUA is LEU. Right, so now it's a different amino acid, right? It's a mistake. So this is called a missense. Missense mutation. No dash. Missense mutation. Now, is this bad? That's the question you have to think about, right? Like, is this bad? Well, it depends on several things, okay? So is this bad? Okay, it depends. Okay? Depends on the location of the mutation. Right? If this was an enzyme, the protein is an enzyme, and the mutation happens right in the active site. The active site is where the ingredient fits in, right? If the mutation happens in the active site and it changes the shape of the active site, then that's pretty bad. Okay, let me try to redraw this for you. Remember enzyme, right? Like, you know, that's going to be the substrate and it's going to fit into the active site. But if the mutation happens in the active site and it causes it to change shape, right? Changes shape to this, then, you know, it will no longer fit, right? The square won't fit anymore. Then the whole enzyme is, you know, useless. So that could be bad. But if the mutation is like elsewhere, it's not on the active site, it's just like somewhere else in a part that's not too significant, then maybe it's okay. So it's going to be dependent on the location. Number two, it's going to dependent on the properties. The property of the amino acid. Long time ago, lecture 2 now, I think. Right? We talk about proteins and amino acids. And I hope you remember the amino acid has side chains, right? Some side chains are acidic, some side chains are basic, some of them are hydrophobic, some of them are hydrophilic. So if you change a hydrophilic amino acid after the mutation and it's also hydrophilic, then maybe it's okay. Maybe the impact is minimal. But if you change it from hydrophilic to hydrophobic, that's going to have a huge impact. If you change it from acidic to acidic, maybe it's okay. But if it's acidic amino acid and then becomes a basic amino acid after the mutation, then that could be pretty bad, right? Okay. So that's all. Any questions about missense mutations? All right. Last one here is called a nonsense mutation. So that's like this. Wild type is, let's say UCA. And then the mutation, the mutant. Now, I cannot stress the fact that this change had to happen in the DNA, okay, in the coding strand. So like technically it's like, you know, TCA, right? But I'm just doing it in the RNA to make it easier for you to understand. So please look up UCA.
SER.
SER, okay. And then mutant, what is UGA?
Stop.
Stop. That's right. So now think of the whole protein, right? The whole messenger RNA, 5' to 3'. Now usually the stop is at the end, right? So you will make the amino acid, and then when you get to the stop codon, it stops. Yeah, that makes sense. But if you change one of the amino acid, the codon, if you change it to stop codon, let's say the stop codon happens here. Let's say you have a nonsense mutation there, then you will make this amino acid 1, 2, 3, 4, 5, and then you stop. Then the rest of the protein is gone. Right? So this is called like when you don't make the rest of the protein, when the protein is shortened, is called a truncated protein, a shortened protein. And again, the question, is this bad? Well, it depends. If it happens early on, that's pretty bad. You lost the whole protein. If it happens towards the end, maybe it's okay. You lose a few, right? So Lika asked how many mutations exist. We are almost there. 3 types for now, and I have 2 more to show you. So does anyone have any questions about the 3 types of mutation so far? A neutral, which is kind of like the best one to have, missense and nonsense. You cannot say one is worse than the other. Okay? It all depends on location. Okay? Location, depending on like where it happens. Right? A nonsense mutation that happens at the end could be a better alternative than a missense mutation that happens in the active site. Okay. So here are some examples to show you. Okay. With the coding strand and the template strand. You can see if you change, let's say you change this one. This one says like GCT. So if you do the transcription, GCT is going to become GCU. Okay. And if you look up GCU, it's going to code for arginine, ARG. But if you change the A to T, so it's no longer CGC. I'm so sorry, I read it wrong. Okay. If you look up CGC, it's going to give you arginine. But if you change the C, right, to A, and you have CAC, then the code is going to be different. Right, so that's a missense mutation. So that this just shows you what happens if you change the DNA, which then the mistake gets carry on when we do RNA, right?
Okay, so the
yeah.
first sorry. Yeah, so this first one is the good one, or like templates.
Yeah, so this one is like this, right? So this is saying, CCT, and then they're saying, okay, let's change it to CCC.
Oh, okay.
CCC like that. Right? So it's good for us, I guess, because it was proline before and now it's still proline, no change. So I think I told you about sickle cell anemia. That's when your hemoglobin is misfolded. The protein hemoglobin helps carry oxygen for you, and then it causes the red cell to go from normal to sickle cell shape, to moon crescent shape. And so the amino acid for the hemoglobin has, I think, 128 amino acid in it, and when number 6 is changed, this is already in the middle of the sequence, okay? When they change it from GAG to GTG, essentially you look up GUG, right? You can see the amino acid goes from GLU, glutamic acid, to valine. And it turns out the original is polar, but then the mutant is non-polar. So that single change in a long sequence of amino acid causes the hemoglobin to misfold, and then it causes the cell to be different. And people with these sickle cells, red cell, they cannot transport oxygen efficiently, and it's going to cause them to experience symptoms like shortness of breath. Okay, they cannot exchange oxygen with the tissue effectively. They might feel fatigue, malaise, and it actually could be painful because these red cells get stuck in capillaries and it, blocks them. So that's an example of a missense mutation that have very dramatic results. Okay, I am sorry. I think I'm gonna have to skip the break again because we still have quite a bit to cover. I do need to finish everything before you do the quiz next week, right? So another kind of mutation that we're going to talk about is insertion and deletion. Okay, so far, the nonsense, missense, and the silent mutation, it's all about changing a single nucleotide, right? Base substitution. But with insertion and deletion, it's exactly what the name sounds like. You are putting extra nucleotides in the sequence, or you're deleting them. Okay? And that could happen, right, during DNA replication. And when you insert or delete nucleotides that are not multiples of 3, you're going to end up changing the reading frame. And we call that a frame shift mutation. Now that is like makes absolutely no sense to you unless you've learned this before, right? So I'm going to give you an example. We know the ribosome reads the code in triplets because 3 nucleotide makes a codon. So you read them in triplets. So it's like this sentence. Okay. The big cat saw the fat dog. That makes sense. But now let's take a look at the next sentence. In the next sentence, I actually deleted the B. Okay. So the first word, it's okay. It's still the. But after I deleted the B, and remember, I can only read 3 letters at a time, the rest no longer makes sense. Okay. And the last one is, just only 2 letters. I can't make out what the sentence is saying anymore for the rest of the way, right? Because by deleting the B, we shift the reading frame. And everything changes after that point. Okay. What's the change here in the next one? Compared to the wild type, compared to the original, what did I do, huh?
You deleted the old BIG.
That's right, deleted BIG, right, which is 3 letters, and that's okay. I mean, I lost a bit of information. I don't know like the size of the cat, but the sentence structure is maintained, right? Like the sentence still makes sense. I can still read it, even though I lost a bit of information. Whereas the previous one, like the whole thing is no longer, legible, right? Same thing here. Right? I've inserted an extra letter. I inserted a K right here. So everything is shifted. But if I insert 3 letters, now I have extra nucleotide that shouldn't be there. I have extra words that shouldn't be there. But again, because I'm inserting multiples of 3, I'm able to maintain the sentence structure. Okay, so whenever you insert things and take things out, that results in what we call a frame shift mutation.
Okay, so in what situation would a new codon be added to the mRNA?
That is a good question. Yeah, so first of all, the change does not happen in the RNA, right? If they're gonna add something, the mistake will have to happen in the DNA level.
DNA, right.
That's right, yeah. So it happens during the DNA replication, right? And it's more common for sections of the DNA where there are a lot of repeats. We'll learn more about this in a biotech unit, but there are some sections in the DNA that just keeps on repeating itself. Like, CGA, 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 repeated like 20 times. So when the DNA polymerase copied that section, sometimes it slips. It miscounted. Instead of copying 20 of these CGA, it accidentally slipped in like an extra one. So instead of 20 CGA, you got like 21 CGA. Okay? And sometimes you might miss one. Okay, so that's where it happens. Most likely is going to happen. Okay? But for inserting like a single nucleotide or missing a nucleotide, that pretty much can happen anywhere. Okay. All right. So I want to go back to the. Give me a second, please. I want to go back to the PowerPoint, the study guide here, and I want to do this question with you. Okay, so this is in the study guide. I'm just putting it here so that I can do it with you. Okay. So this is the original DNA sequence, and the question actually tells you that this is the template. The template. Now, for the interest of time, I mean, I think you guys already know how to convert by this point. So I'm just going to quickly do it for you. Okay? It's better to chop it up into triplets so we don't, miscount ourselves. We don't sometimes get the wrong letters. So because this is template, the mRNA is going to be complementary, right? And, I'm going to put a dash here to separate it for me so I don't get confused. So nothing is new here. I am totally just doing the complementary rule and writing out the mRNA sequence. Okay? And then using the codon chart, I'm going to translate it. And again, I'm assuming that you already know how to do that. And we learned it last class, and we had some practice up here. So I think that's a reasonable assumption. So you look up AUG, and that's going to be MET, TRP, ASN, ARG, CYS, and stop. Okay, so this is the original DNA sequence. Another name for original is wild type. It's what happens in the wild. Okay, so let's take a look at our first mutated DNA sequence. We're going to chop them up into triplets. And we see that it's, evenly chop out. I don't have like an extra thing or missing a letter. It's the same length, in other words. So I know that this is not insertion. This is not deletion. This is just a base substitution. So you compare the letters, the nucleotides one by one. Tell me where the mutation is happening. Where's the mutation?
On the second part of the 3 kill.
I'm sorry, which one is this? Is this the first set, the second set, third set? Which set?
The second one.
Second one. And what's the change?
And it was ATC instead of ACC.
That's right. So the C becomes the T, right? That's the change. That is a base substitution. Okay. So I'm gonna copy that. I'm gonna change it to mRNA. No change for the first one. And then the second one, it's ATC, right? So when you change it to RNA, it's gonna be UAG. Okay, and then the rest are gonna be the same. Okay, I'm not gonna copy everything. What will the amino acid sequence be? Well, the first one is still AUG, isn't it? So AUG, if you look it up, I mean, you should memorize it by now, right? The start codon is always MET. And then what's UAG? Someone tell me what UAG is, please. That's right, Leah. It's the stop codon. UAG is the stop codon. What kind of mutation is this? What do we call this?
Nonsense.
This is nonsense. Such a fitting word, right? This is nonsense. Nonsense. Okay. Will there likely be an effect? Yes. The whole protein is gone. The whole protein is missing. You bet there's going to be an effect. Right? So I should probably expand this a little bit. This is a base substitution. And then it leads to a nonsense. I just like to put a dash there, like it just looks better for me. Some people just
Professor,
don't use it. It's
excuse
fine.
me.
Yeah, go ahead.
When you select the mutated DNA sequence, for example, T, you already circle it.
Mhm.
And then can we explain on there, please?
Can I
This
explain?
T, yes.
Okay.
How we identify the
Oh, the
mutated,
change?
yes.
Yeah. So look, TAC, TAC. So that's fine. And then ACC, but now this is ATC. Right?
Mm.
So you just compare.
Yes, yeah, I got it. Okay,
Okay, good.
yeah.
Anyone else? All right. Let's just do one more. So we you know, there are lots. There are like 5 of them, and it's the answers in the study guide answers that I posted. But let's just do one more. Okay, so I'm gonna chop this one up into triplets. Always chop it up into triplets. Okay, and now you can see, like, you know, there's an extra one there, right? So probably somewhere along the way, there is an insertion mutation. So we have to identify it. TAC, TAC, no change. Next is ACC, but now I see GAC, right? So in fact, the ACC is there, but then there's an extra G. So that's where the mutation is. Okay. So I'm going to write down that this is an insertion. Insertion of the G, right? So let's change it into messenger RNA, AUG, CUG, GAA, so on and so forth. Yes, go ahead.
Excuse me, Professor, I didn't understand why TAC is not, how to know that TAC is not changed. It's because of the T is at the last of the,
How do I know TAC is not changed? Is that your question?
yes,
Yeah, so TAC, you have to compare to the original, right? Original is TAC and this is still TAC, right?
but we compare all of them to the original.
Yeah, in this particular question, this is the wild type, and then these are like different mutations of the original.
Okay, now I got it. Thank you, Professor.
Yeah, sorry for the confusion. So we're gonna look up AUG, which is MET. We're gonna look up CUG, right? CUG, what is CUG? CUG is LEU. Okay, GLU. I'm just looking it up, okay? And then dot. So, what happened? Well, the insertion of the G actually led to a frame shift. Led to a frame shift, which then results in many missense mutations. Okay. Missense just means it's a mistake, right? It's wrong, right? In fact, everything after where the G was inserted, everything after that are wrong. All of them are wrong. Okay. So will there be an effect? Yes. The original protein is gone. You're not even making the original protein. It's the wrong recipe. It's like you're supposed to make a cake, but now, you know, you're making a chicken instead. Like completely different. All the amino acids are different. And the worst part is like it doesn't even stop, it just keeps on going. Right? Who knows when is it gonna stop? Yes, this is part of the quiz next week. Yeah. The quiz is 6 and 7. 6 and 7. Okay. But like I said, the quiz is gonna you're gonna have the quiz available from Wednesday to Friday. Okay. Wednesday to Friday.
Professor, excuse me, the PowerPoint, I could not find the PowerPoint, so I was confused.
You cannot find a
For today's
PowerPoint?
for today's lecture.
Anyone else have a problem with that? Just curious.
Just in lecture 6 and 7, in lecture 7 is protein synthesis, so there's
Yeah.
no other PowerPoint.
Hold on. Anyone else have trouble with the thing? Okay, this is the PowerPoint link. All right. Sometimes you might not be able to see it until you download it. Okay, depending on like your browser setting. So like once you get to it, like if it's showing nothing, then you click on the download button. Okay. And again, things are a little bit weird on the phone sometimes. So like, you know, if you're not seeing it on the phone, make sure you try it on the computer. Okay, so you can try more later on your own, but that's pretty much it for now. Okay, we're gonna go back to our PowerPoint here, and we will do a few more.
Professor, 55 slide, there is protein synthesis.
Say
Is
that one more
correct?
time.
55 slide.
55? Yeah, 55 slides, that's right. Okay. Yeah,
Okay.
so yeah, okay, you got it, right?
Okay, Professor,
Yeah.
thank you.
So remember the you know, I should talk about this before I forget. The lab is gonna be like you have to download the lab report, just click on the link, and then you're gonna submit it there. And again, I cannot open Mac computer file, so please make sure you submit it in PDF format or .doc format. There are some calculations in the lab, but you know, I create a video to help you with those calculations, so make sure you watch it before you do it. Okay, even if you think you understand how to do it, maybe watch it just in case. All right. Quiz 3, which includes lecture 6 and 7, is going to be available from Wednesday to Friday. So you know, if you are not ready to do it on Wednesday, don't rush it. Study more first. It will be available to you until Friday 11:59 p.m. Okay. 30 questions, 60 minutes. Okay.
Vincent,
Yeah.
when you get the chance, could you please post the notes from last week, if we have one?
The class notes from last week.
Yes, please.
Did we have any notes last class? Did I not post it? I don't. Hold on, let me see. Class notes. Last week was what? March, 13 and 11. Okay, yeah, I'll double check. I don't know if we have any. Maybe we didn't. Oh, did we? Oh, yeah, we did. March, which one is this? March 13. Yeah, we just had one small one. That's all. Okay, but sure, I'll post it. No problem. Okay, back to here. Back to the PowerPoint. Okay, so here it says, now this one you don't have to worry as much. Okay, it's more for my in-person class. Right? For my in-person class, I'm gonna ask them to create mutations on the test. But for you guys, like for the online environment, it's hard for me to ask these kind of questions. So don't get too hung up with this thing next. But I'll show you how to do it anyways. Create a base substitution in the coding strand that will result in a missense mutation. So this is coding. So all I have to do is make a messenger RNA. Okay, and you can choose any of them. Let's say I start with this one. Let's say I choose this one. This is AAG. So if this is coding, then the messenger RNA is also AAG, right? So I want a missense mutation. The easiest way to do it is just change the first letter. Okay, so if I change the first letter to like C or something, CAG, right? Change the first letter to C, then it's a missense mutation. We can look up AAG. That's going to be LYS. And then we look up CAG. CAG. That's going to be GLN. Right? So that's how you create a missense mutation. You just change the first letter, basically.
Each of them, Professor?
Not all of them. You just have to do one, right? Because it says,
But
yeah, it
whatever
says
we want, put the difference.
Yeah. I mean, like, okay. So let's do another example, right? Let's say you take this one. Okay. This one is GAT, right? So even after you change it to RNA, it's going to be GAU. And then you can change it to like CAU. And that's going to be another missense mutation, right?
Okay, thank you.
Yeah. So changing the first letter, in fact, changing the first or second is almost always going to be a missense mutation. Okay, here. Create a base substitution in the coding strand that will result in a nonsense mutation. So nonsense, right? So it's probably a good idea to change the whole thing to RNA first so that we can see it better. I mean, this is coding, so it's very easy. You just copy it. So we need it to be a nonsense mutation, which means I need to have stop codon. And you know the stop codons, UGA, UAA, UAG. Told you last week, right? So where can I create a stop codon? Well, over here, if I change CAG, if I change it to UAG, that's a stop codon, right? But remember, I must reflect the change in the DNA. So it would have been a T right there. So that's going to be a nonsense mutation. And you basically are going to be missing the whole protein. Huh? Can someone give me another place where I can make a nonsense mutation here, please? Where else? It's a little bit challenging, huh? Okay, so for example, I can do it right here. This here says AAG, but if I change it to UAG, right, that's another stop codon, right? Okay. So you can either do it in the second one or the third one. Right? Both is going to create a nonsense mutation. Okay, you don't have to do both. You just have to choose one.
Vincent, why are you changing the DNA, the strand, the coding strand?
Yeah, because remember, in reality, the mutation is not happening at the RNA level, right?
Yeah, it's happening. Oh, okay.
That's
yeah.
why, yeah. Because if you have a change in the RNA, theoretically that could happen. If you have a change in the RNA, then the problem is not going to be permanent because the RNA doesn't last forever. Remember? Right? So if the mistake is in the RNA, as soon as the RNA breaks down, then you are okay. But if it changes in the DNA in the original, then every time you copy it, you're still gonna have that mutation, right? Okay, let's do another one here. Number 3, create a base substitution in the template strand that will create a nonsense mutation. So again, we are going to just copy, just make it into mRNA first to make it easier. But remember, this is the template, so we have to use complementary rule. Okay. I'm just changing from template to mRNA. There's nothing special about this. And now I have to create a stop codon because it's asking me for nonsense. So UGA, UAA, UAG. Now let's try this again. Tell me where can I make the change so that I'm gonna have a stop codon.
The AAG?
Yeah, we can change the AAG. We can change it to UAG. Okay. So in the original, because this is the template strand, the U would have been paired with an A, right? So we have to flip it. You can do it here as well. Instead of UUA, we can change it to UAA. That's another stop codon. And on the original, it would have been like that. Okay, so there are multiple ways this could happen. In a way, it's kind of scary because mutation can happen like anywhere along the way, and it could really mess up the protein. Okay, last one here. Create an insertion mutation in the coding strand that will result in a truncated protein. So this is back to coding, which is great because I can just copy it. Now I need a stop codon. Okay, so where do I insert an extra letter to create a stop codon? Okay, so let's read it, right? Like AUG and then GAA. But what if I stick in a U there? If I stick in a U there, then the ribosome is going to read AUG, UGA, and that's a stop codon. So you can create stop codon by base substitution, but you can also create stop codon by insertion. Okay, and then so that means in the original, I'm going to have to insert a T. Okay. So I know that last 4 examples here, it can be a little bit confusing. I'm not saying, like don't worry, don't care about this at all. Like you can go through this yourself and it really would add to your understanding of what I'm trying to teach you. But don't worry about answering these kind of questions on the test, on the quiz, okay? Like I cannot ask you to like show me a mutation, right? Like physically it's hard for you to type in the answer for something like that, right? So in terms of a multiple choice question, I won't be able to ask you these kind of questions. Okay, so maybe there is a little bit of comfort knowing that. Okay, let's wrap this up. Which of the following mutation is likely to be the most deleterious? Deleterious means very harmful. So let's see. Deletion of AAA at the beginning of a gene. Well, because we are deleting 3 things at a time, there will be no frame shift. No frame shift. We will be missing an amino acid. Nah, that probably is not the worst thing that could happen. Okay. Next one. UAC is mutated into UAU in the middle of a gene. So let's quickly look up UAC and UAU. UAC is TYR. UAU is also TYR. So that means this is just a neutral mutation. And we know neutral mutations are the best kind to have. If you're going to have a mutation at all, you probably want to have a neutral one. It's not going to do anything to you, right? Okay. UAC is mutated into UAG. Well, this is a nonsense mutation, but it's happening near the end of the gene. So you are not losing a whole chunk of the protein, right? You might lose a few amino acids, and maybe that's okay. Right? So far, like this is probably the worst out of the 3, but let's read on. Deletion of an A nucleotide, as in like an adenine, at the beginning of the gene. Well, if we delete something at the beginning, then you're gonna have frame shift. Right? Remember, like deleting the letter B, the whole sentence structure is gone. You're gonna have a lot of missense, many missense. All right, so that's probably pretty bad. Okay, so I'm gonna keep this one on my radar, and then just read the last one. Insertion of UAA at the end of a gene. Well, UAA is a stop codon, but again, if you're doing it at the end, maybe it's okay because you're gonna stop anyways, right? If you stop a few amino acid earlier, maybe it's okay. So this is the worst one. All right. That's it. Okay, so no class on Wednesday. Just focus on finishing your lab. And the lab's going to be due Friday, right? Yeah. So, you know, if you want to hand it in early, sure, no problem. Excuse me. But, you know, you have until Friday. And if you need help with the lab, if you're like not sure what the question is asking, feel free to email me. And also, right, understand that there is the quiz to do. So no class on Wednesday. Next time we have class will be on the 25th. All right. Well, thank you for sticking around. Super long lecture, I understand. And have a good week. All right. Good luck with that stuff.
Teacher,
Yeah, go ahead.
is this possible to extend the quiz time frame because the lecture is so big? Is it possible to extend till Saturday? Actually,
Yeah.
so complicated and so big.
Yeah, I guess. Sure. Yeah, it's but I mean, it's already 3 days, right? But sure, I'll extend it to Saturday. It's fine. Yeah, no worries.
Thank you, because it's so complicated and so big.
No, I know.
Oh,
So much.
perfect. Thank you.
Okay, well, take care, guys.
Thank you.
Thank you.
Bye-bye.
Yeah, bye.
Professor.